Today we will discuss about a new paper where they talk about this immunotherapeutic approach against the ovarian cancer using a IL-12 based nanoparticles. So their idea is that they took a nanoparticle based drug delivery mechanism for immunotherapeutic treatment against the ovarian cancer and then optimize that nanoparticles so that it will have a efficient response against the ovarian cancer. So let us understand how they did this. So they did this liposome, and then they did the layering it with different steps. So first they coated it with this interleukin 12, and once the coating is done with interleukin 12, they coated it with this positive ions, that is poly-L-arginine, which is a net positive, which will give a net positive charge on this, and then they coated it with this poly-l-glutamate, which will give them a net negative charge. So in this stage they check whether their layered or coated nanoparticles performance against the conventional nanoparticles towards targeting this ovarian cancer. So in this figure we are not discussing about the ovarian cancer's results. We will discuss about its efficacies in terms of this interleukin 12 exposures and its effect with the cancer cells. So first what they did is they check, and in this case the one important thing that they mentioned is the solvent in which these lipid nanoparticles or their coated nanoparticles are kept. So when there is no, for storage purpose they kept them in the DI water. So we will understand why they kept it in the DI water and what are the changes occurs when they move them into the serum based media. So first they check the effect of this serum based media on this coated nanoparticles, that this PLE and PLR that are coated on this liposome will remain on it even in this physiological media, or they will get removed. So first they check it in the serum based media. So in the serum based media, when they check them, they are able to see that around 40% of this PLE and PLR are removed, and also this around 60% of these PLEs are removed when they move their coated nanoparticles into the serum based media. Thereafter they also wanted to check the exposure of this interleukin 12 in the case of this DiH2O solvent, or when they move them into this 10% FPS containing media or the serum based media. So when they did it for the conventional here they did not did it for the conventional. Here they shows the result for the DiH2O when the coated nanoparticles are in this DiH2O, and when it is in the serum. In the DiH2O they are able to see that they did shows the results for the conventional also. For the conventional they considered the reference of this interleukin 2 expressions is as 1, and here they did the ELISA to check how much interleukin 12 are been attached to the plate. So for the conventional, in the DI water they are able to see his signals, that is considered as a 1, and when they did it for their coated nanoparticles, the condition of the amount of IL-2 attached to this ELISA plates are lower. When they move them into the serum based media, they are able to see some of the recovery of this attachment, some of the recovery or increased binding of the coated nanoparticles onto the ELISA plates. So this indicates that the serum plays an important role for these IL-2 exposures. And thereafter they check this IL-2 retained, how much IL-2s are retained. They also wanted to check whether these IL-2s are also removed in the serum based media or not. So here they did the studies. Here they did it not here they did the studies on this DI water, where they see that in the case of this DI water, and they're coated. So this is not in the DI water, these are in the 10% FBS. In the case of 10% FBS, they checked that the IL-2 removals for the uncoated nanoparticles is high, whereas IL-2 removals for the coated nanoparticles, that is their optimized nanoparticles, is lower, indicating that it will have a higher lifetime within the patient. Thereafter they wanted to check, when they co-cultured them with the cells, whether they are able to interact with the cells or not. So for that they took one of the cell lines. I'm not able to recall that cell lines, but it is some kind of a cancer cell lines. And when they did co-culture them with these lipid nanoparticles and then check the exposure of the nanoparticles, in this case they use the fluorescence based nanoparticles. So they are able to see that around some portions of the cells are attached, some portions of the lipid nanoparticles are attached to the cells in the when they use the uncoated lipid nanoparticles, but the signals from the coated lipid nanoparticles attached to the cells is significantly higher. And these things they have quantified in the panel F also, where we can see that the increase in the case where they use layer by layer coated lipid nanoparticles, or this liposomes, which are after coating with different reagents such as interleukin-12, PLR, PLE, gives a high binding or high attachment with the cells against which these are co-cultured with, against the cells in which these nanoparticles are co-cultured with. Then they use the confocal imaging to find out where these lipid nanoparticles gets into the cells. So when they check it for the uncoated lipid nanoparticles, they are able to see that most of them are internalized within the cells. Whereas for the case of this, so these are the signals for the cells that can be, these are the signals for the nanoparticles within the cells. And for the bottom row, where they shows the result for the coated nanoparticles, they are able to see that these the coated nanoparticles, the nanoparticles are localized into the membrane of the cells, that can be seen here. So this indicates that since the nanoparticles are localized to the membranes, there are high chances that they will play a role towards cancer signalings, cancer death signalings, or cancer suppression signalings. So far they have just showed that the efficacies of their modified or optimized nanoparticles using interleukin-12 has seems to be better performance, seems to be have better properties compared to the conventional nanoparticles that is used for this ovarian cancer immunotherapeutic treatment. So that's all for this video. I hope you understood how they optimize their conventional nanoparticles to improve their therapeutic approach or therapeutic treatment against the ovarian cancer. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.